Okay, so we are doing the topic biological molecules, which is chapter 4 from your textbook, and today we are going to cover proteins in chapter. So you may keep the page numbers 45 open with you, which is chapter 4.4 about proteins. Okay, so let's start with the verse from the Quran about the expansion of the universe, which says بَنَيْنَاهَا بِأَيْدِي وَإِنَّا لَمُوسِعُونَ. Okay, so it says, and it is we who have constructed the heaven with might, and verily it is we who are steadily expanding it. Subhanallah, we discussed about it already. The objectives for today's topic would be to explain the properties and functions of proteins. Alright, because we already discussed the properties and functions of carbohydrates in our last topic. And fats we are going to discuss after this. Alright, so today we are going to do chapter 4.4, which is proteins. Okay, under the KWL, let me ask you a few quick questions about what we covered yesterday. So we discovered, we were studying about the sources and the structure and also the classification of carbohydrates yesterday, right? So can you please give me a few sources of carbohydrates? From where do we get the carbohydrates? From the food we eat?
Cereal and wheat.
Very good. Cereals. Wheat is one of the cereals. And?
Vegetables and bread.
Bread and what did you say? Bread and?
Vegetables.
I'm sorry?
Vegetables.
Vegetables. Okay, vegetables are not a very good source of carbohydrate except for the fiber which we get from vegetables. Okay, yes, so that would be a correct answer. Very good. Fiber we get, which is a source of carbohydrate from the vegetables and the fruit skins, and all the wheat products which we eat, like the bread, the khubz, the spaghetti, the pasta, all these are made up of wheat, and all the baked foods which we eat, which are made up of refined flour. Okay, and all the cereals, rice, millets, maize, all the corn products, all these would be the sources of the carbohydrates. The most common and abundant sources are sugars, fibers, and starches. So what is the simplest unit of carbohydrates?
Sugar.
Sugar, very good. Glucose is the simplest unit of carbohydrates. And yesterday we also discussed about the classification of carbohydrates. So how can carbohydrates be classified into? What are they classified into?
Monosaccharides, disaccharides, and polysaccharides.
Excellent. Can someone explain what are monosaccharides with examples?
Yes, can I?
Go ahead, sweetie.
Monosaccharides are the molecules that has a single carbon, and
single carbon?
Yeah, single carbon ring.
Okay, can we say single carbon? Yes, we can say single carbon ring. That is also correct, and single sugar molecule also can be used. Excellent. Examples?
Examples can be glucose and galactose.
Very good. Yeah, thank you so much, sweetie. And who's going to explain what are disaccharides?
Can I explain this?
Go ahead.
Disaccharides, they consist of two sugar molecules, and some examples include sucrose, lactose, and maltose.
Excellent. Thank you so much. And who's going to explain polysaccharides? Quickly.
Miss, can I?
Go ahead, Laila.
There are many sugar molecules, and for example, maybe like starch.
And what are the other examples? Many sugar molecules, how are they linked together?
Like cellulose also.
Very good. Starch, cellulose, and glycogen, yes. And how are they linked together, the sugar molecules and the polysaccharide structure? They are linked together in the form of a long chain or a branch chain, right? like we discussed, these would be the structure or this is the glucose molecules are linked in the form of unbranched chain in cellulose and branched chains in glycogen and starch. Thank you so much, Laila. Okay, so that was the classification of carbohydrates classified into mono, di, and polysaccharides. Can someone please explain the difference between respiration and breathing? The girls who didn't get a chance to speak so far. Habiba, can you please explain it to me? No answer. All right. Anybody else? Please raise your hands who wants to explain. Quickly. Ahead. Go ahead, sweetie. Ahead.
Sorry, the mic was not working. Respiration is the metabolic reaction that happens, but breathing is just the exchange of gases in our body.
Excellent. And what do we get after the process of respiration? What does the body get?
Gets like what? Miss, I don't understand the question.
Okay, sweetie. After the process of respiration, what are the end products?
The end products will be carbon dioxide and water.
Very good. And what is released during this process?
Released, like it's glucose and oxygen.
Yes, sweetie. The glucose is broken down once we take in oxygen. The oxygen combines with the glucose in our body, and of course, carbon dioxide and water are released as waste products, and energy is also produced or released during this process, isn't it? ATP is also produced. Okay, so you can put this equation as glucose plus oxygen gives rise to carbon dioxide plus water plus energy in the form of ATP, which the body uses for various daily activities. Excellent. All right, that was about carbohydrates we discussed so far. I also assigned you the homework. You may submit it over the weekend whenever you're free. So there are two presentations over here. This is a presentation of paper 4 styled questions, okay, which you didn't do so far. All right, so these would be structured questions. I'm going to explain in detail as to how you will answer these questions. So they would be more than 2 to 3 line questions like this. I've uploaded this presentation on Teams and I'm going to explain it to you next week, inshallah, once we are done with the chapter 4. All right, so this is one of the questions from paper 4 will be included for your midterm exam. Okay, the others would be MCQ questions from paper 2, and only one structured question from paper 4 will be included for your midterm exam. All right, sweeties. So we shall go through all these questions from this presentation so that you have an idea as to how to answer it. Okay, and this is a presentation yesterday we were doing on the food test. So let's also revise about the food test which we do in order to check the presence of starch and glucose in the food. All right, so name the food tests which are used for glucose and starch. Which are the food tests we studied about yesterday?
Iodine testing.
Very good, iodine testing. So what do we do and what happens in iodine testing?
Can I
you
answer
read
that?
out from the slides, sweetie. Yeah, go ahead.
Do I explain or do I read this?
Yeah, anything. You may explain it. That's better.
Okay, so if when we use iodine testing to test whether starch is present in certain foods or not, and if the iodine turns yellow or if it turns blue-black, then the starch is present, therefore it's a positive test. But if it still remains yellow after applying it on the food itself, then it's a negative testing, meaning that starch is not present.
Excellent, sweetie. Very well explained, Shahab. So do we need to apply heat for this test? Is heat applied?
I don't think so, Miss.
No, heat is not applied. Very good. For this test is direct. You may just inject the iodine into a potato or any food containing starch, and immediately the iodine changes color from yellow to blue black. So if it changes color, meaning that particular food, it contains starch. Whatever you're checking. So heat is not applied in this test. And who is going to explain the test used in order to check the presence of sugars in the food? Quickly. Who wants to explain it? Sumaya, would you like to do it? Shardon, Rimas. Quickly, sweeties, we are running out of time. Jana Jamil, would you like to do it, sweetie? No one. Jana?
Yes.
You want to explain what is Benedict's test?
No.
Great. Okay, let me do it then. So Benedict test is a test which is used in order to check the presence of sugars. We are normally testing the presence of simple sugars like either monosaccharides or disaccharides. Okay, so Benedict's test identifies the reducing sugars. Reducing sugars meaning they are either monosaccharides or disaccharides which have free ketone or aldehyde functional groups. Okay, so Benedict's solution can be used to test for the presence of glucose in the urine as well. So this is how the test looks like. Benedict's solution is actually blue in color, and whenever you're adding the solution to it which contains glucose, okay, so it gradually changes color from blue to brick red. Okay, so a positive test meaning the color of the Benedict solution changes first from green to yellow to orange to finally brick red. A negative test meaning if the food does not contain glucose and it stays blue. Alright, so in this test, heat is also applied. You need to also apply heat over here. Okay, so this is what we discussed even from your textbook. Benedict solution to we add the Benedict solution to the food and we heat it. And if the food contains the reducing sugar, then the brick red color will be produced. Okay, so this is how you will do it. You will be doing these experiments, inshallah, in the lab. So what are the safety precautions you need to follow when you're doing these lab experiments? Do you need to wear an apron and goggles? Do you need to wear goggles throughout? Why?
For your safety.
Because of the chemicals, mixing of chemicals.
Yes, very good. So you get many fumes and smokes whenever you're heating, especially, and because you're dealing with many chemicals. It's always safe for you to follow the safety precautions. So you have to wear an apron or a white lab coat and gloves always, and also goggles so that your eyes will not get affected. And also if you have any particular allergies to any kinds of smells, you may also use a nose mask, the mask which we are wearing these days. So that also can be used in the lab. And you need to be very, very careful when you're dealing with the beakers and the test tubes and all, because they are made up of glass. And also take care, particularly when you're heating and wherever hot water is involved. And especially because you're doing these tests on the food, always be away from the food in the labs. Do not try to eat it. Do not get tempted to eat, because those foods which you are using are being tested there. They are not kept there for you to eat. All right, so don't eat the food when you're doing this lab test. All right. These are a few questions which you will be able to answer today only about the carbohydrates, because we covered only till carbohydrates. So we shall answer these questions after we complete the subtopics of proteins and lipids. All right. So let's go back to our presentation today and talk about proteins. We are on page number 46 from your textbook. Okay, and let's talk about proteins in detail now. Now, proteins are large complex molecules that play many critical roles in the body. So may I know what are the functions of protein in our body, girls?
It helps with the building of muscles.
It helps for the what of muscles?
The building.
Building of the muscles, excellent. And
For repairing cells.
for repairing cells, very good. And what else are proteins used for in the body? They are used for building up of the cells. They are used for repair of the worn out cells and damaged cells. And what is the primary function of proteins in the body? Quickly. Any other functions you can think of? Yeah, Mariam is raising the hand. Go ahead, sweetie. Mariam, she put her hand down again. I don't know why.
Enzymes?
Yes. Could you repeat that?
It builds enzymes, or it builds them up.
Very good. It helps in building enzymes in the body. Enzymes which are present in the body are definitely proteins. Excellent. By the way, congratulations, Mariam, for receiving the recognition Student of the Month. All right, that's it. Okay, yes. So you skipped important function of proteins, that is growth. Okay, so the primary function of the protein till the growing stage, that is for you, you are in the growing stage right now, till the age of 18 to 20, proteins are primarily used in the body for growth. After the function of growth has been done, that is after you cross the age of 20, for your lifetime till you die, the proteins are used in your body for repair and maintenance. Very good. Proteins are used for the buildup of the cells. They are used for the as you answered, they are used for the repair of the damaged and worn out cells in the body. They are also used in the making of antibodies, and the various enzymes present in the body are made up of proteins. Okay. So proteins, they do most of the work in the cells, and they are required for the structure, function and regulation of the body's tissues and organs. So why are they saying they are required for structure, function and regulation? Is proteins, they are the ones which are the building blocks of the cells, and of course the cells, they come together to form tissues and organs, and that is why they form the structure, function and regulation of the body's tissues and organs. And they are made up of carbon, hydrogen and oxygen, exactly like carbohydrates, but they also contain two other elements which are nitrogen and sulfur. All right, so this is how a structure of a protein molecule looks like, as you can see in this picture. Apart from carbon, hydrogen, oxygen, the protein structure also contains nitrogen and sulfur. All right, so this is how the structure of a protein molecule looks like. They show hundreds or thousands of smaller units called amino acids, which are attached to one another in long chains. Okay, so exactly like polysaccharides, in polysaccharides, the various glucose molecules are linked together in the form of long chains. Similarly, the protein molecule, it is made up of amino acids which are linked together in the form of long chains. So there are 20 different types of amino acids that can be combined to make a protein molecule. All right, and the sequence of the amino acids, it determines each protein's unique three-dimensional structure and its specific function. All right, depending upon the arrangement of the amino acids, the structure and the function of that particular protein molecule is decided. Each and every protein molecule has a different structure, and depending upon the structure, that is the combination of the amino acids, the function of the protein, it also differs. All right, clear so far, ladies? Clear about the structure of the protein molecule? Any questions? Shall we move on?
Yes, Mas.
Thank you. All right. So you can see, like polysaccharides, protein molecules are made up of long chains of smaller molecules joined end to end, like this. Okay, so these smaller molecules are called amino acids. There are about 20 different kinds of amino acids, and any of these 20, they can be joined together in any order to make a protein molecule. So these 20 amino acids, they can be combined with any different combination or any order to make the structure of a protein molecule. And each protein, it is made up of molecules with amino acids in a precise order. Even a small difference in the order of amino acids makes a different protein. All right, so each and every protein, it has a unique structure which is unique to itself, which defines even the function of that particular protein. So there are millions of different proteins which could be made. So you can see one of the structure of the protein molecule over here, where you can see the presence of the amino group and the carboxyl group over here. Okay, so this is a structure of one of the protein molecules. Okay, so now coming up to the enzymes. We already know that even proteins are enzymes, right? And the different sequences of amino acids give different shapes to protein molecules. All right, so not only the function of the protein molecule depends on the combination of amino acids, but also the sequence of the amino acids gives different shape to the protein molecules, as you can see in this picture here. So these different shapes of protein molecules can be related to their function. Enzymes are proteins that provide a surface for reactions to take place called the active site. Okay, so these enzymes, they have various functions in our body. They help in aiding digestion and many, especially the metabolic reactions which take place during the digestion in our body, they need enzymes. Right? For example, the salivary amylase, which is present in our saliva, the pepsin, which is released in our stomach, and various other enzymes which are released in our body in order to carry out these metabolic reactions. So enzymes, they act as catalysts in these reactions. So do you understand? What do you understand by the term catalyst?
They help quicken up processes.
Yes, very good. They help in speeding up the reaction or quickening the reaction. The rate of the reaction is increased wherever enzymes are involved. But all the enzymes, they cannot be used for any or all the metabolic reactions in our body. A specific enzyme can work only on a specific active site. Okay, meaning how the enzyme works depends on the lock and key mechanism. Like a particular key only fits, or only you can use that particular key in order to open the lock. It depends on the dents which are present on the key, which are exactly similar to the dents present inside the lock. So can you use any key to open any lock? No, isn't it? Similarly, the enzymes are also the proteins that provide a surface for the reactions to take place called the active site. As you can see, this enzyme has this shape of the active site, and only the substrate can, only this shape substrate can fit into these enzymes, and only when the substrate fits, the enzyme substrate complex is formed and the enzyme can start functioning. Okay, so only only a few substrates act along with the enzymes, or we can say enzymes, they form an active site only for a few substrates, and when these two come together, only then the enzymes, they can function. All right. Okay, now let's talk about antibodies. What are the antibodies? What do you understand by the term antibodies? Okay. Antibodies are also proteins which are produced in our body, but these are produced in order to provide defense in our body. Okay, we already know the WBC, with the white blood cells in the body, they have the function of, they are known as the soldiers of the body, right? They help in defending us from various pathogens or foreign particles which enter our body, isn't it? And these antibodies are proteins which are released by the WBC, or they are also proteins that bind along with the pathogens, and they help in killing or destroying the pathogens. Okay, so antibodies are proteins with a structure that has binding site on its surface like this, and this enables them to bind with the chemicals called antigens. Now, what are antigens? Antigens are the protective coat which are present on the top of the pathogens. For example, virus, they are covered by an outer protective coat which is known as a capsid. And on top of the capsid it is covered, it contains various surface proteins which helps the virus to get attached to the various cells. Okay, so this uniform which is present on top of these pathogens like virus and bacteria, it is known as an antigen. Alright, so these antibodies, what do? Whenever there is an pathogen which enters our body, they bind themselves to this antigen on the surface of the pathogens and make the pathogen stick together. Now, what is the function, or what happens when these all pathogens stick together is what we are going to watch in the video further. I'm going to show a video. Now, these pathogens, they all stick together, and all of them, they are engulfed by a type of a WBC known as phagocytes. Okay, so you will be studying in detail about the types of WBC which are present in your body and their function under the immune system. Okay, so for now you need to know that there are 6 different types of WBC cells which are present in your body, each one of which has a specific function. Out of them, 2 of the cells, they are given the name phagocytes. Phagocytes, the term phago meaning they can engulf, that is swallow the pathogen, be it a virus or a bacteria or fungus, and they can destroy it. Okay, I will explain that to you with the help of a video and a short now within a few moments. Before that, let me also quickly cover the information given in the textbook. Okay, ladies, so please open page number 45 from your textbook. Let's go through the information from the textbook as well. Any questions?
I'm sorry to interrupt you, but can you please send us the highlighted parts after?
Yeah, I shall do that.
Okay, thank you.
You're most welcome. So protein molecules, they contain some kinds of atoms which carbohydrates and fats do not. What are they? The types of atoms which are present only in proteins? They are nitrogen and sulfur. So as well as carbon, hydrogen and oxygen, they also contain nitrogen and small amounts of sulfur. You can see the picture in your textbook. Like polysaccharides, protein molecules are made up of long chains of smaller molecules joined end to end. And these smaller molecules are called amino acids. And there are about 20 different kinds of amino acids, and any of these 20, they can be joined together in any order to make a protein molecule. And each protein, it is made up of molecules with amino acids in a precise order, and even a small difference in the order of the amino acids makes a different protein. Now, coming to the functions of proteins, some proteins are soluble in water. An example is hemoglobin, the red pigment in blood. Okay, so the red color pigment which gives the blood its red color, hemoglobin, even this, it is a protein. Okay, and this hemoglobin, it can be soluble in water. You can see that whenever you are whenever you're losing blood, you must observe that the blood, it can mix with water. Or whenever you are trying to wash off the blood, you can see that it is soluble in water. Others are insoluble in water. For example, keratin, the protein with which the hair and the fingernails are made up of. Okay, so if you have observed our fingernails, our hair, you know how the structure looks like, a little tough. Okay, so hair and fingernails are made up of a protein known as keratin, and of course it is not soluble in water. Is it soluble? No. Right, you know how hard and tough the nails are when you're cutting them, and also your hair, how it looks like. Of course, it is not soluble in water. So unlike carbohydrates, proteins are not normally used to provide energy, and many of the proteins in the food you eat are used for making new cells. So new cells are needed for growing and for repairing damaged parts of the body. In particular, cell membranes and cytoplasm contain a lot of protein. And proteins are also needed to make antibodies, as we are discussing right now. And these help to kill the bacteria and viruses inside the body. And even enzymes are also proteins. So the long chains of amino acids from which protein molecules are formed can curl up into different shapes. The way in which the chain curls up, and therefore the three-dimensional shape of the protein molecule, is determined by the sequence of amino acids in the chain. So different sequences of amino acids result in different shapes of the protein molecule. Are you following, girls? Is it clear about the structure and function of proteins?
Yes, miss.
Any questions so far? No questions? All clear?
Yes, miss.
Thank you so much. All right, so can you see the picture you have on page number 46? This picture, this is a model of an enzyme called lysozyme, all right, which is found in the saliva and the tears. So this particular enzyme, it is found in the saliva, in your saliva, and also in your tears. The name of the enzyme is lysozyme. So the purple band represents the chain of amino acids which is coiled up to produce a small depression called the active site. Can you see the purple color of this enzyme in this picture? So that is the chain of the amino acids, and it also has an active site in it, right? So small depression called the active site, and the yellow part is another molecule which is a substrate molecule that fits perfectly into this active site. Okay, so these enzymes, they have a particular shaped active sites, and another substrate, it needs to fit exactly into that enzyme site so that this particular enzyme can work. So this is what I was telling you about the lock and key mechanism. So over here, the lysozyme enzyme, it is the lock, and the yellow color substrate, it needs to fit into this lock in order for that lysozyme enzyme to work. Okay, so for most protein molecules, their shape directly affects their function. For example, you will be talking about a lot of protein molecules now in the next chapter, which is enzymes. They are going to talk about enzymes, how do they work, and all that in your chapter 5, inshallah. So how the enzymes act as catalysts. So the shape of the enzyme molecule, it determines which reactions it can catalyze. Similarly, the shape of an antibody molecule determines the kinds of bacteria or viruses that it can attach to. Okay, so I'll just show you a video now as to how the antibodies work. Now, different shapes of antibody molecules are needed to bind to different kinds of bacteria and viruses, and each different kind of antibody therefore has a different sequence of amino acids from which it is built. Okay, so how the antibodies work in your body? Let's watch this video. Do let me know if the audio is clear.
A foreign bacteria has invaded. One way the body...
Is the audio clear or no?
Yes, Miss.
Yes, it's clear.
All right. Let me play it again.
A foreign bacteria has invaded. One way the body protects itself is with small proteins called antibodies. These Y-shaped molecules recognize the intruder and bind to its surface at special sites called epitopes. When antibodies begin to cover the bacteria, it is called opsonization. This process attracts a phagocyte, a large cell that digests unwanted particles in the body. The phagocyte binds to the antibodies and engulfs the bacteria. The microbe is then killed and digested, and the body saved.
Did you understand as to how it happens? Did you get it or no? Can someone explain what actually happened in this video? Okay, let me show it to you again. See. Okay, so this green color thing, which is
a foreign bacteria has invaded.
It has
one way the body protects itself is with small proteins called antibodies.
So these are the antibodies.
These Y-shaped molecules recognize the intruder and bind to its surface at special sites called epitopes.
Antigen. Now this outer coating of the bacteria which you see is known as the antigen. And these antibodies, they fit themselves to the outer coating of the antigen. So that is what they were explaining about how the antibodies function. Different shapes of the antibody molecules are needed to bind to different kinds of bacteria and viruses. Okay, so each and every body, it binds to the different shapes of the antigens, that is the outer covering of the bacteria or virus, which enter your body and they start acting upon them. Then what happens?
When antibodies begin to cover the bacteria, it is called opsonization. This process attracts a phagocyte, a large cell that digests.
Okay, now these antibodies, they help in attacking the bacteria. Sometimes they help in stopping the action of the bacteria, and sometimes they bring all such type of bacteria in one group. Okay, in one clump, they try bringing all the bacteria together. This helps these phagocytes. The phagocyte which you are seeing, which is coming and swallowing this bacteria, it is the kind of WBC. It is a phagocyte which swallows this bacteria, which engulfs it and kills it.
Unwanted particles in the body. The phagocyte binds to the antibodies and engulfs the bacteria. The microbe is then killed and digested, and the body saved.
Did you understand now how the antibodies and the phagocytes, they work in killing the bacteria?
Yes, Miss.
Yes, is it clear, ladies? Any questions? You need to tell me whether it is clear or no. Is it clear? Any questions so far regarding the proteins topic?
Yes, Miss.
Eva? Yeah, Maria. Clear, Maria?
Yes, Miss.
No questions? All right, so this is about the topic proteins. We shall discuss about the testing for proteins in our next lesson, inshallah. In the same presentation, the next we have the testing as to which tests are done in order to check the presence of proteins in the food. So the test for proteins, it is known as a Burette test. Inshallah, we'll discuss in our next lesson. We don't have time now. Let me assign you the homework. The questions you have on page number 46, that is from 4.10 to 4.14, will be the homework for the proteins topic, inshallah. So you need not worry about submitting the homework now. You must submit it over the weekend or until the end of next week whenever you get time, because I understand that your midterms are commencing now and you have a lot to study. So whenever you get free time or whenever next week or any time, you may submit the homework. No hurry at all. Okay, ladies? In the next lesson, inshallah, I shall go through the paper, 4 questions for you. And if you have any questions regarding the midterm content also, you may ask me that day. Ahead, what is your question, sweetie?
Miss, I wanted to ask for the unit test. Are we going to study unit 1 too?
No, you have. I sent you the midterm content on the group, isn't it?
Yes, Miss.
It's chapter 2 and 3.
We will not study 1?
No, not required. Only the cells, the chapter 2 and chapter 3 will be covered.
Okay, thank you.
Okay. Osmosis, diffusion, active transport, which is movement in and out of the cells, and cell structure and organelles. I clearly mentioned in the midterm content, so only that you have to study. All right? Okay, then you have a great day ahead and